In today's video, we're going to be looking at all of these different points that we need to know for the AS Biology Paper 3 exam. If you want to skip to a certain point that you see on this paper, take a look in the description below as I've listed the timestamps. So let's get started. The independent variable is the variable that we control. It's the thing that we are changing. On the other hand, the dependent variable is the thing that we're measuring, and it changes as a result of our independent variable, right? It's dependent of our independent variable. A control test is a test that we do where we keep the independent variable constant or neutral. A standardized variable is a variable that we keep the same throughout all of our tests. Now I'll give an example that includes all of these different variables. Let's say, for example, we want to measure the effective pH on the rate of enzyme activity. Our independent variable, well, we're going to have different pHs. The dependent variable, what are we measuring? Well, we're measuring the rate of enzyme activity at those different pHs. A control test, so what can we keep neutral? How can we neutralize the independent variable? Well, we could do a test with a solution where the pH is 7, where it's neutral. Or we could use water, for example, which has a pH of 7. A standardized variable would be the temperature, for example, because pH affects enzyme activity, so does temperature. So if we want to see only the effective pH on the enzyme activity, well, we'll want to keep the temperature the same throughout all tests. We have two types of dilutions. We have a simple or proportional dilution, and we have serial dilutions. Simple/proportional dilutions are dilutions that we do where between each successive dilution we are decreasing the concentration by a constant. Okay, so let's say, for example, we wanted to make successive dilutions and reduce the concentration by 2%, by a constant of 2%. So we'd start off with a concentration of 10%. The second would be 8%. The third would be 6%. The fourth would be 4%. And the fifth concentration would be 2%. On the other hand, a serial dilution is where we reduce the concentration by a factor. Okay, so let's say we started out with a concentration of 10% and we wanted to reduce the concentration by a factor of 2 between each successive dilution. So the second dilution would be 5%, the third would be 2.5%, the fourth would be 1.25%, and the final one would be 0.625%. So those would be our dilutions. We can see that. So we're using a constant of 2, right? We're reducing the concentration by a constant of 2 between each successive dilution. That's a simple or proportional dilution. And for the serial dilutions, we're reducing the concentration of each successive dilution by a factor of 2, right? We're halving the concentration each time. Here we've got 5 tests. So what that means is whenever we do a procedure, unless it's otherwise specified, we want to have 5 different independent variables, 5 values for our independent variable. So if, for example, we want to measure the pH, measure the effect of the pH on an enzyme's activity, right, we would have 5 different pHs. If we wanted to have 5 different concentrations of a certain solution, we'd have 5 different concentrations, right? 5 types of the independent variable. We've then got 80 degrees Celsius to 100 degrees Celsius. And what this means, so if we're boiling water, if we want water to be boiling, once it reaches 80 degrees, we classify it as boiling. If in a procedure, it asks us, so if in a paper, it asks us to modify a procedure, and in our modified procedure, we need to boil water, well, we're going to have to state a value in between this interval, like 85 degrees Celsius, for example, in order for the water to be boiling, right? We cannot say 80 degrees Celsius to 100 degrees Celsius. No, we say a value in between that interval. Now, moving on to sources of error. So the first thing to understand is that a source of error is not a mistake, but instead it's an inaccuracy in measurement, right? It's something that makes our results invalid or unreliable. Two types of errors. The first one is a random error, which by chance gives us a different observation than the true value. On the other hand, a systematic error is an error that we do throughout all tests, and that's going to skew our data in a certain direction. For example, here where you see this point, this is what our true value should be. If we make a systematic error, instead of our true value being at that point that we have, all of our data is going to be skewed in one direction. Two examples of a source of error are non-precise measurement intervals. For example, we have to measure 3.6 milliliters of a solution, and our syringe only has intervals 1, 2, 3, 4. It's going to be quite difficult to exactly measure 3.6 milliliters. Another example is a non-exact independent variable. So for example, if we are trying to measure the effect of changing the temperature on enzyme activity, and we have temperatures listed 10 degrees, 20 degrees, 30 degrees, 40 degrees, and 50 degrees, but when we're actually testing, we have 12 degrees, we have 24 degrees, we have 28 degrees, 43 degrees, etc. Those are non-exact independent variables, and that's going to give us invalid data. So 4 methods that we can do to fix sources of error, right? And whenever we are asked in the paper to find sources of error and state how we could improve them, these are 4 things that we should always firstly think about. So the first one is to use smaller intervals for values of the independent variable. So for example, we're measuring the effective temperature on enzyme activity, and we have 10 degrees, 30 degrees, 50 degrees, 70 degrees, and 90 degrees. Well, if we want to get more accurate and more reliable data, we could look and see. So if we know that the optimum temperature for that enzyme is, let's say, 40 degrees, well, we could bring the interval and make it smaller, and we could say, okay, let's choose temperatures 20 degrees, 30 degrees, 40, 50, and 60, right? And that way we can get more finer details and we can get more reliable results. A second way that we can fix a source of error is to measure the dependent variable more accurately. So if, for example, we have a test tube with a solution and we have a syringe with another solution, and we're pushing the syringe and we're pushing out liquid into the solution that's in the test tube, and we're counting the number of drops of solution from the syringe that it takes in order to make the solution in the test tube change color, that might be unreliable because each drop might not be the same size. So a way that we can better measure the dependent variable is to measure the amount of solution required to make that color change occur, right? The volume, we can measure the volume, the difference, so we can measure the initial volume that we had in the syringe and take that away from the final volume that we had in the syringe, and that would give us a more accurate measurement, right? Instead of counting the numbers of drops, we just measure the amount of solution that we had to use in order for the color change to occur. The third thing that we could do is we could carry out repeats to calculate an average or mean. So we can do this to eliminate any anomalous results, right? We can use this, so we can carry out 3 repeats of each test in order to obtain a mean, and that's going to give us more confidence in our results, right? If it happens 3 times, it means that it's more likely it's true. Whereas if it happens only once, well, we're not entirely sure. Maybe we did something wrong. If we do it 3 times, well, then we're going to eliminate the error and we're going to be able to get the true, or we're going to be able to get more accurate results. The final thing that we could do is we could find a way to standardize important variables. For example, in the enzyme, if we want to measure the rate of enzyme activity at varying temperatures, right? Something that we're going to want to keep constant is the pH, because pH directly affects enzyme activity. So throughout each test, we're going to want to have the same pH so that we're only seeing the effect of temperature, of changing the temperature. So that's it for this video. I do hope you enjoyed, and if you found it helpful, take a look at some of my other videos that might interest you.